All right, so I'll also be sharing my screen with you as we proceed in the course of this class. Okay. Okay. Now, okay, let me just share the screen right away. Can you see my screen now?
Yes.
Okay, good. We're going to be looking at biological molecules, and we have defined biological molecules are chemical substances that make up the cells. Biological molecules are chemical substances that make up the cells. And each of these chemical substances are vital for life. They are vital for life. Each of them are vital for life. Okay. Now, when we talk about biological molecules, we are looking at carbohydrates, fats, protein, and of course, in some instances, nucleic acid is also included. In some instances, nucleic acid is also included. Okay. Now, for what you're seeing on my screen, you see biological molecules, and then you also see the elements that is present in each biological molecule. We have carbon, hydrogen, and oxygen to be present in all the three. Carbon, hydrogen, oxygen is present in carbohydrate, in fat, and in protein. But then protein have additional nitrogen and sometimes sulfur. So it has nitrogen and sulfur. So protein has excess of nitrogen and sulfur. Okay. So just know that the most common elements in the three biological molecules that are listed here are carbon, hydrogen, and oxygen. Now, these biological molecules are found in food that we eat, all right? The biological molecules are the kind of food, they are found in food that we eat, okay? And we have different test methods to know which particular biological molecule is present in which kind of food. Okay, now look at the table. You can see there's a table there, testing for biological molecules in food substance. We are testing for starch. We are testing for reducing sugars, like sugar, that's like glucose. We're testing for protein, and we are testing for fats. So we have food that contains starch, food that have glucose, that contains protein and fat. Now, the name of the test for starch is called a starch test. All right. And the reagent, I'll call it the reagent or the method that is used. Okay, in the method, you see the reagent actually. So we're going to read the methods to you now. From the method, you see the reagent. Now, what is the method? Add few drops of iodine solution. Iodine solution is usually brown in color to a solution of the food. Okay, we're looking at starch in a particular food. So what do? We make a solution of the food and then add iodine solution in it. How do you make solution of the food? By turning the food, maybe it's adding some water to make it becomes liquid. You make the food become liquid by adding some water, and then you add iodine solution. Okay. Now, how do you now know what is the test result? The positive test result tells you that iodine, the food itself, the solution of the food, would change. The color of the solution would change from brown to blue black. From brown to blue black. Blue black. Not blue or black. It's not, the answer is not blue or black. It's blue black. It's actually supposed to be blue black. Bluish black. See? So That is a test. I want to know, can you hear me? What was the last thing I said? Paul, what was the last thing I said? If you can hear me. Bayan, you can hear me.
That's it's bluish black.
Okay, good. So when you mix up the solution of iodine plus food substance that contain starch, they become brown. But the brown solution of iodine turns the solution into brown, but the result will show blue-black after a period of time. You see that the color will change from brown to blue-black. Okay, the test for reducing sugar is called Benedict's test. Benedict's test. Benedict test. So how do you do that? Add an equal amount of Benedict solution. Now, Benedict solution is the reagent. And the first one, iodine solution, is the reagent. Reagents are used, the name I usually call reagents that are used to test for biological compounds, in food or other substances. So add an equal amount of Benedict solution, which is usually light blue. Benedict solution is usually light blue, to a solution of the food. Then what do? You boil it. After you've added it, the solution has been mixed together, and then you boil it for some minutes, like 10 minutes. You boil it for like 10 minutes. Then you will see that the blue solution will change from blue to green to orange and to brick red. Now, if the color change to green, it means that the glucose in the food is very small, very minute, not much. If it changes to orange, it means the presence of glucose in the food is medium, like average, medium, which means it's more than the green. So the orange is more than the green, so it's medium. But when it changes to brick red, it means that there is a lot of glucose in the food that we're testing. Okay. The next is a test for protein. The name of the test is Burette test. Burette test. You can see the spelling on my screen. This is it here. This is Burette test. How do you do it? Add Burette solution, usually light blue, to the solution of food. Add Burette solution, usually light blue, to a solution of food. And the blue solution of food will turn from light blue to violet. Violet is like, what color? Like purple. Call it like purple. Violet looking like purple, but it's actually violet. Violet is the color. So it changed from blue, light blue, to violet. That is the test for protein. The test for fats is called emulsion test. Emulsion test. How do you do it? You dissolve the food in ethanol. Ethanol is a kind of alcohol. It's a kind of alcohol. So you dissolve the food that you want to test in ethanol. Then pour the solution into a clean test tube of water. When you do so, you will see that after a period of time, there is a white emulsion on the beneath the solution. You see white emulsion. See, now that white cloudy white emulsion in the test tube is indicative of the presence of fat. It indicates that there is fat in the food that you are testing. Okay, I hope you can hear me clearly, everything I've said.
Yes.
Okay. Now, next, we will look at the structure of protein. Now, the drawing you are seeing here represents what I will call polypeptide. We call it polypeptide. Why? Because there are several amino acids, and you can see that the color of the different balls, the colors are not the same. They are different colors. Why? Because these different balls represent different types of amino acid. Now, remember, we have 20 different types of amino acid in the whole world, the universe. 20 types of amino acids that code for every kind of protein that is in the world. So only 20. So when you have several amino acids comes together, depending on the number that is being put together, it forms polypeptide. So this thing you're seeing here, this long chain, is a polypeptide chain. Polypeptide chain. A bond, this chemical bond between this amino acid and this amino acid is called peptide bond. Even though you call it chemical bond here, the chemical bond, the name of the chemical bond is peptide. Peptide is spelled like this. Let me see if I can put it here. Peptide. Peptide. You can see it as a peptide bond. That's the name of the bond. This chemical bond is called peptide bond. Okay, and each of these is amino acid. And of course, you know what amino acid is. Amino acid is the basic functional unit of protein. The smallest basic functional unit of protein is amino acid, which means after the last digestive, when you break down protein to the barest minimum or to the smallest particles, you get to what? Amino acid. And you know, during digestion of protein, it is the end product of such digestion of protein is amino acid. Okay. Amino acid is what is assimilated into the body for proper function. So you can say, let me read this. Okay, maybe somebody should read. Okay, or Bayan, can you hear me, Bayan? Are you there? Can you read the first paragraph, Bayan? All right, if Bayan is not hearing or cannot read, I would like Ho to read the first paragraph.
Okay, mister. The chain of amino acid in a protein takes up a particular shape as a result of cross-linkages. Cross-linkages form between amino acids that are not neighbors, as the shape of protein molecules have a very important effect on its reaction with substance.
Continue. Okay, just hold on. Let me read that. Now, from what you have read, the chain of amino acid in a protein takes up a particular shape as a result of cross-linkages. And cross-linkages form between amino acids that are not neighbors, as the shape of protein molecule has a very important effect on its reactions with substances. Okay? It has a very important effect on its reaction with substances, which means different chains of amino acids are usually brought together and that form the different shapes. Now, for an amino acid, it has three kind of molecules that forms the amino acid. We have the amine group, we call it the amine group. We have the carboxylic group, and we have the variable group. The variable group is actually where different kinds of amino acid can be attached or bonded with the structure of amino acid. Okay, so an amino acid structure has the carboxylic group, and then has the amine group, and then the R group. The R group is the variable where the different amino acids, like I said, we have 20 kinds of amino acids. So the variable says that any of these 20 can be attached here. And when it's attached there, it means that, okay, that is the name of the particular protein. Okay, the name that the protein bears, depending on the kind of protein. If it is like a monomer of protein, like one single amino acid, then it means that the amine, this is called the amino acid. You get it? So, but then when it's added with the other ones, the other variable, then it takes the name of the variable that's added. Okay, but that is actually not for your level. It's a higher level. But the only thing you should know is what is written here. Just know that the chain of amino acid in a protein takes up particular shape as a result of cross linkages. So the shape of the different amino acids are not the same. Every amino acid have different kind of shape depending on the variable, the type of amino acid that is cross-linked with in the amino acid chemical structure. Okay. Antibodies are protein. We talked about antibodies today. So we say antibodies are proteins produced by white blood cells called lymphocytes. Antibodies are proteins produced by white blood cells called lymphocytes. Each antibody has a binding site, which can lock onto pathogens such as bacteria. This destroys the pathogens directly or makes it so that it can be detected. It marks it so that it can be detected by other white blood cells called phagocytes. And of course, you know what phagocytes are. Phagocytes are antibodies that engulfs or eat bacteria, eats pathogens, and digest them. Each pathogen has antigens on its surface. Each pathogen has antigens on its surface that are a particular shape, so specific antibodies with complementary shapes to the antigens are needed. So that's what we call the antigen antibody or antibody antigen, complex. We have the enzyme substrate complex. We also have the antibodies antigen complex. Okay, so antibodies generally are protein, and of course we know that proteins are biological molecules. So we're looking at different kinds of protein produced by white blood cells, called lymphocytes. And then we also say that there are two kinds of antibodies here. We have the phagocytes, and then we have the lymphocytes. Okay, and that each antibody has what? A binding site. Each antibody has a binding site. Okay, where we have pathogens such as bacteria can lock up, where they can be locked up or lock into, okay, and destroy. Because when they lock a particular bacteria as an antibody, they end up destroying the pathogens. See, how do they destroy the pathogen? By eating them up or by secreting chemicals to destroy them. Okay, I think that's all. Then we look at the importance of water as a solvent. What are the importance of water as a solvent? Okay, somebody can read that now. Importance of water as a solvent.
Do I read, Mr.?
Yes, you can. Read. It makes it fast.
Importance of water as a solvent. Most cells contains about 75% of water. Water helps in digestions and excretions. Many important metabolic reaction takes place in aqueous solution. Many substance moves around a cell dissolves in water and also around organism, example in blood, xylem and phloem.
Okay, very good. Importance of water as a solvent. Most cells contain about 75% of water. All the cells in your body, they are in liquid state. Most of them, they're in liquid state because most of the part of the cell is 75% water. Water helps digestion. Without proper intake of water, you have challenge with digestion of food. And in excretion, because without water, we can't be able to excrete waste product. Okay? Of course, some of those are also excreted as metabolic waste. Some of the water are excreted as metabolic waste, but they're very important to create a better environment for digestion and excretion to take place. Okay, many important metabolic reactions take place in aqueous solution as well. There are several reactions that cannot take place in a place that is too cold or that is too hot. Many substances move around a cell dissolved in water and also around organisms, e.g. in blood, xylem, and phloem. Okay, so those are the importance of water. Okay, now we're going to look at the next one, which is called the ribonucleic acid. Firstly, if you look at the name, deoxyribonucleic acid, when you see the word nucleic is the same as nucleus. So DNA is found in a nucleus. That's why it's called nucleic acid. So DNA is an acid that is found in the nucleus. Don't forget that. DNA is an acid that is found in the nucleus. Okay. Now, aside from that, the smallest breakdown, just like I say, the smallest breakdown for carbohydrate is glucose, that's the smallest monomer of carbohydrate. The smallest monomer of protein is amino acid. The smallest monomer of lipids are fatty acid and glycerol. The smallest monomer of DNA or nucleic acid is what? Nucleotides. Nucleotides are the smallest unit. See, and usually DNA always have two strands. Two strands. You can see the strand here, the first strand and the second strand. So they have two strands that are wound together. They are wounded together, like in the shape of a double helix. Like the shape of a double helix, they are wounded together. That is a DNA. And of course, DNA is where all the genetic information of an organism is inscribed. It is in the DNA. See, everything about an organism, humans, plants, animals, is in the DNA. Okay, so if you have access to someone's DNA or an animal's DNA or a plant DNA, then you can reproduce that kind of plant again, or reproduce that kind of animal again, if you have the access to the DNA of such organism. Okay, so what are the nucleotides? In DNA, the sugar is deoxyribose. That's why it's called DNA, deoxyribose nucleic acid. So it is without oxygen, and the organic base are either A, T, C, and G, where A is adenine. Now, these adenine, thymine, cytosine, and guanine are called bases, nitrogenous base. They are called nitrogenous base. Nitrogenous base. Let me write it here. They are called nitrogenous base. They are called nitrogenous base. So A, T, C, G. These are nitrogenous base. And these are the nucleotides that are being used to synthesize nucleic acid, that are being used to transcribe to form a particular protein, like an enzyme or hormone. All right? And they have their different codes. Okay? So you can see it here. This is cytosine, guanine, adenine, thymine, nitrogenous base. So, okay, we have 10 more minutes to finish this class. So, every DNA has nitrogenous base, A, C, G, T. A stands for adenine, T stands for thymine, C stands for cytosine, G stands for guanine. And then, of course, we have two kinds of nucleic acid. There are two types of nucleic acid. We have the DNA and we have the RNA. DNA and RNA. DNA is double-stranded. It has two strands. RNA has only one strand. For the nucleotide of DNA, it has cytosine, guanine, adenine, and thymine. For the nucleotides, the nucleotides of RNA, it has cytosine, guanine, adenine, and uracil. So if you look at it too, you notice that they're the same. The only difference is in thymine. DNA has thymine and RNA has uracil. But others is the same. Adenine, guanine, cytosine, adenine, guanine, cytosine, the same. But the last one of it is uracil. So how do I know that a particular strand is RNA when I see the uracil? I know it's RNA. How do I know that a particular strand is DNA when I see that the strands are double? There are two, wound together, and as thymine. If there's thymine, then it is DNA. If there's uracil, then it is RNA. So those two bases or nitrogenous bases are what we use in identifying which particular nucleic acid is this. Is it a DNA? Is it an RNA? Okay, so That is it. Now, if you look at the complementary base pair, you see base pair here. In setting up on the sequencing of nucleotides, like in a double strand. In a double strand, you have a nucleotide at the one strand and another complementary nucleotide at the second strand. At the one side and the second strand. At the one side and the second strand. So the two sides have a nitrogenous base. Okay. Now, how is it pairing goes? This is a pairing. Let me give it to you here. I'll be to write. Well, okay. Let's do it there here. Okay, in the pairing A, okay, is here. A always bind with T for DNA. For DNA, this is DNA. Oh, okay, let me just go here. This is DNA. So A binds with T and C binds. Now C has triple bond, not double. It has triple bond. I don't know. I can't do that here now. I don't know how to insert the second, the other bond, but is a triple bond. It has triple bond with G. So it has triple bond with G, while A and T has what? Double bond. In RNA, it's a lot different. Well, almost similar, but also different in the area of, okay. So in the RNA, we have A bonded with what? Tell me what it will bond with. Tell me. And then C bonds with G. So tell me what this will bond with for RNA. Or
With
anyway,
U?
correct. It will bond with U, uracil. So that is the difference between the two. Okay? All right, so I'm done. You'll do the worksheet 4.1 and then you bring to school on Sunday so I can see it. Worksheet 4.1. Okay?
Okay, Mister.
Do
Thank
you have
you.
any question?
No, Mister.
You
Thank you.
sure? Bayan,
Yes.
do you have any question?
Bayan left the meeting.
Okay, she left the meeting. So you explain to the two of them, please. All right?
Yes, Mister.
Okay. Thank you so much.
Yeah.
Before you sign out, let me see something first.
Yes.
I hope I've been able to cover everything. So if I ask you, for instance, what are the nitrogenous bases? What can you say about them? What are they? Can you list the nitrogenous base?
They're adenine, thymine,
Yes.
cytosine, and the G one.
Yeah, the guanine.
Guanine. Yeah.
Guanine. We call it the guanine. Okay. So, and if I ask
Yeah.
you, what are the complementary base pairing? What can you say about that?
DNA
Yes.
and RNA.
Yes. What is the complementary base pairing? What are the bases that pair with which one?
A for DNA, A and T,
Correct.
and for RNA, A and U,
Okay.
then G and C
For both of them.
for the
For
yeah.
RNA and for DNA. Very good. So you're good.
Yeah.
So what is the test for starch?
We put an iodine solution. If it turns into bluish black, then it shows that presence of starch.
Very good. What about the test for fats?
What, Mister?
What is the test for fats? Lipids?
Just a second. For what, Mister, you said?
How do you test for lipids or fats, as the case may be?
We add an equal amount of Benedict solution
No,
to a solution of
no. Benedict solution is for glucose. What is the test for fats? Fat or lipids?
fats. Dissolve the food in ethanol. Then we
Yes,
pour a solution into a clean test tube of water.
yes. And then
Then
what
it
is
will turn the positive result will turn into white emulsion.
Okay, very good. That's good. All right, thank you so
Yeah.
much for joining. Do have a good weekend. All right,
Thank
bye.
you.